Ah, there, I got it. Good. All right. Okay, can somebody confirm you can hear me?
I can hear you.
Great. Before we begin, are there any questions? All right, let's go to the syllabus. So today is July 30th, almost the end of July, and we are going to discuss and start the Unknown Project. Hopefully you've taken a look at the instructions for the Unknown Project. This is the identification of an unknown bacteria. It's a 75-point lab project, and if you were to come into an actual lab, microbiology lab, I would give you a test tube that had two different bacteria species in it. And then you would streak it out and streak it out for colony isolation and get an isolated colony and streak that out for on another plate to get a second time colony isolation so that you send your cells through two separate isolated colony isolation events so that you'll get a pure culture. You then take the cells from one colony and start your stock culture. And in the future, you would do all your work on the stock culture. So you would keep your stock culture alive and use your stock culture to run all your biological tests with. Now, obviously, we are not in the lab, and everything we do will be done online. So we're going to mimic that, where you're going to pretend to streak out a culture and isolate cells and get a pure culture, and then pretend to use these cells to set up your different biological tests. And you're going to run different biological tests to try and identify this species of bacteria. And what I'm going to do is send you a number that will represent a species of bacteria, and you will run the different tests to try and identify that species of bacteria. Is that clear to everyone? So you're not actually going to be working with bacteria and trying to identify the species of bacteria. I'm going to give you a number, which is going to represent a bacteria species. And then you're going to run different tests to try and identify that species. So these are the species that the bacteria might possibly be right here and right there. Scroll down a little bit. One of those species. And then these are the different tests that you can run up on the top here. There are a few other tests you might be able to run, but these are the tests you can get information on. And then you use these tests to try and identify the species. Now, initially, when we were thinking about this project and doing it online, because of COVID-19, we had to move all classes online for the first time. We came up with this procedure. And it occurred to me that if you were to ask for each test and then contact me with the test and then send it through, you would be doing on average 5 or 6 different tests. And then I would be replying 5 or 6 times to each student. For one class, that would be 20 different students, which would be a minimum of 100 different contacts between me and each student for each class. So that would be 100 different contacts for one project. And I just decided that that was too much for each student to have 100 times where we were contacting each other for one project. And so I spoke to the different instructors and I said, how about instead of them picking the first 3 tests, I pick the first 3 tests, and then I will reduce the number of contacts by 60. I think that's per student too, but something like that. Maybe it's 30 per student, something like that. I forgot the exact numbers, but the point is, and they said fine, and then the students can pick the rest of the tests to identify the species. And the only reason why we did that was 100 different contacts on average between each student and the professor. It's just too much for one project because with 100 different contacts with 20 different students, that's an awful lot of different contacts. So I'm going to pick the first 3 tests for you to run. And instead of sending it out as 3 different messages, I'll send them together. And so instead of having, I'll reduce the number of contacts by 60. That means I'll still have 40 different contacts with each student for one project, but that's doable. The 3 tests I've chosen that I will do will be the gelatin test, actually the gram stain test, the gelatins test, and the PR lactose test. The gram stain test is just a good one to run because it'll divide the possible species down to half of them. You'll divide the species either into gram negative cells or gram positive cells down here. And then the reason for selecting the gelatin test is because this test takes at least a week to run. And so it's a good thing to get this test done early because later, if you're running out of time and you have a test that takes a week, you're going to lose a lot of time waiting for that test. This test needs to incubate for a week before you can get the results. And then the PR lactose test is just a good test to run for narrowing down the different species. So let's say we have gram negative species. That narrows it down from all the possibilities to about half of them, shown in yellow. And then we're going to go the gelatin negative species, which are here and here and here. And then we do the PR lactose test and it turns out to be AG. It will narrow it down to these 4. And then once you've got it narrowed down to these 4, you look on the chart to determine which test to run next. A bad test to run would be this one here, because all the data is ND for not determined. Another bad test would be the pigment test, because they're all the same. They're all negative for pigment. The same with the oxidase. A possible good test to run might be indole test or a methyl red test. It may be the VP test. And if you're lucky, you could do the citrate test that narrows it down to either 1 or to 3. Okay, so you look at the table and then decide which test to run, and then you request that test to me. And after the bacteria incubate, I will send you a picture of the test that you ask for. And let's say you ask for the citrate test and it's citrate positive, that narrows it down to 3. I chose that one just because it's 3. And then we continue, go further, and then you could do the VP test, for example. And if it's VP positive, you have to go with the plus and the V for variable, meaning a V means you have to take both a plus and a minus as possibilities. Okay, and so you have to run one more test. That's not necessarily the best way to go, but I'm showing you that's one way to go. Anyways, that's what you'll be doing. Any question about any of that? So you'll need this chart here of the bacteria species and then their results. I think I call it unknown list. Let me take a look here. Under I believe it's lab week 5.
For us, it says it's the spring 2019 version. Is that the one we'll be using?
I probably I haven't looked here. You're talking about this right here? And what did I
Yes.
have there when I showed you here? Yeah, spring 2019. Meaning we just haven't updated this table from 2019. That's all it means. So you'll need that table and then the instructions, which I'll talk about here. You'll also need, let's go back to the labs. You'll also need the sample journal for an example of a lab report. It's not going to show well here. Let me get it from here. This is actually a lab report turned in by Travis Kubota, which is an instructor, and he actually wrote this in 2004. And we're giving it as an example for a lab report. You'll want to look at this before you write up your lab report. You don't need to really work with it until that time. It wouldn't hurt to look at it earlier, but you need to look at it before you write your lab report. And what else do we have? Building a dichotomous game plan. If you know what a dichotomous game plan is, you don't even need this. But if you're a little bit confused about what a dichotomous game plan is and how to do this project, I've got this page here. Building a dichotomous game plan. Essentially, what you're going to be doing is you're going to be selecting one test at a time to break your species into about one of 2 possible choices. And then you get the results. And then you take another test, which will be 1 test at a time, breaking your choices into about 2 choices. And this goes through building a dichotomous game plan, some questions to do the procedure. Why build a dichotomous game plan? It's an easy way to identify a species. Rules for a dichotomous game plan. Every test should be dichotomous, either plus or minus. And then an example of a dichotomous game plan. So if you need this page is available to you. If you're perfectly familiar with what a dichotomous game plan is, and you're perfectly comfortable with it, you do not need to use this page. It's for those people who are uncomfortable and they'd like an example. So we have all possible species A, B, C, and D. We have a choice. We say, is the species rectangular or not rectangular? And you could say maybe a rod or not a rod. And species A and B is rectangular. Species C and D is not rectangular. And then we scratch out the non-rectangular, and then we have species A and B, and is the species not white or is the species white? And there's your dichotomous game plan. And then we've identified it as either species A and B here. Okay, any question about that? All right, so you can work through this example. You do not need to answer the questions and turn them in, but you may if you want. You won't get any credit for it, but you may work on that. Like I said, you'll need the unknown list, and then you'll need the unknown report, the unknown notes page. In your unknown notes page, it tells you what to do. Record your daily activities, especially the test requests here. I will provide you with the test results here as well. Record your results and conclusions here. And let me add something here that's missing. And that is only write in the document you upload. Do not use the comments. It is for my use. And if you put something in the comment, I'm going to delete it. So don't use the comments. After I send you the first results, which will be the results for the gram stain of your unknown bacteria, which is really your unknown number, and the gelatin hydrolysis test, and the PR lactose test, you should put your results in the unknown notes pages. And then you should look at the unknown list here to figure out which test to run. And in your unknown notes page, at the bottom of the page, or it could be at the top of the page, put your test request. If you put it in the middle of the page, I will not be looking at the middle of the page every day. I may only look at the middle of the page every week. And so put it at the end of the page or at the front of the page, and I will catch your test request. If you put it in the middle of the document, I may not catch your test request for a week. Because each day I'll be looking, not each day, but about every other day, I'll be looking at the unknown notes page. And if a student requests a test, it has to be at the end of the unknown notes page or at the beginning of the unknown notes page, I will send you that test request after it has incubated. So with the unknown notes page, you need to put your test results and your conclusions for the first 3 tests I send you, and then put your test request in your unknown notes page. I will read the test request, incubate the bacteria, run the test, incubate the bacteria, and then send you a picture of the results. You'll not get the results right away. The test needs to incubate. And I'll send that to you in the comment section, which is why I do not want you to use the comment section. put all of your comments in the document. You should, in the unknown notes page, have the test results and the test conclusions, as well as the species under consideration. Anything you need to comment, put in here as well. Any question about the unknown notes pages?
Yes, I have a question.
Uh-huh.
Do you want one continued document, or do you want us to upload a new document for each time we request a new test? Or do you want it to be like a continuous document and just add it to it?
You can do it either way you wish. If you do make it a continuous document and you write the unknown notes pages formally, you can just copy that continuous document and then use it as a large portion of your unknown report that you'll need to turn in at the end. So that's one reason to make it a continuous document. However, you do not need to do it that way. You may only have the test request you want, and then the results from your last test and your conclusions and the species under consideration up to that point. And that's all you need to have for that one document. And you can also write the unknown notes pages informally. But if you do it formally, then like I said, you can just copy your unknown notes pages and you have to make it one document. And then it'll be a large portion of your unknown report, which is the 75-point document for the project. Okay, either way is good.
I have
And
a
that
few
tells you
questions.
just one more thing, and that is you can upload an infinite amount of documents here, so that's not a problem. I've never had a student run out of space, never had a student need more space, and I've had students upload nothing, and obviously they got very low on this project. So you can put nothing or you can put as many documents as you want. Okay, what was your question?
Okay, so each student will get different bacteria, unknown bacteria, or the whole class gets the same, I guess, examples or
It's
species.
saying each student will get a different bacteria species. It'd be kind of pointless to send the entire class the same species. Then you guys would just share your results and your conclusions and write the same report. No, you'll have a different species.
Okay, I'm looking at the instructions. So when it says phase one isolation, so in this document where you're the unknown notes, we email you asking you basically to isolate it and you start doing the gram and positive smear. So it's a back and forth. If I'm understanding this right?
Yeah, and the document here, let's go to that. Remember I said you're going to pretend to do some things? We're not actually going to do them because this is an online class. If you were in a real lab, you would streak out the bacteria and send it through 2 colony isolation events, and then take the cells from one colony that went through 2 colony isolation events to set up your pure culture. You're going to pretend to do that in this online class. You're not really going to do it. You're just going to pretend to do it. Okay?
Yes.
That's why I say virtually streak out for isolation from a broth culture. Okay? Another difference that's done in the online class, which isn't terribly important, and I think I've got it mentioned here, is I'm only going to give you one number. So in effect, at the very start of this project, you have only one species. If you were to come into a real microbiology lab, I'll give you a tube with 2 species in it. And the only reason why I'm mentioning that is in the sample journal. Let me go to that now. I don't have it up anymore, I don't think. In the sample journal, the instructor is streaking the cells out for colony isolation, and you can wrote that you're pretending to do that, okay? But my point is that you're not really going to be doing that. And so the very start of this, in a sense, you're kind of skipping, and that is because you're getting a peer culture from the start. And we just started with the online class that we would send you just one number, and then you have that peer culture to work with at the start. But you should pretend to streak it out. And you can pretend to streak it out on the 3 media plates, which are mentioned here. Let me see if I can remember not EMB, McConkey's plate and the phenyl ethyl alcohol plate and the TSA plate. Is that confusing? Like I said, we just decided to make it a little easier for the online class, and you're going to get a peer culture to start with, but you should pretend to streak it out, okay? And we're just pretending a lot of things. Like you're not really going to be giving a bacteria, you're going to be giving a number, which is going to represent a species. And the number will be representing one of these species here. And you won't be able to know from the number. You're going to have to determine the species from the different tests that you run. Any question about any of that? All right, let's go on with the instructions. So you can read about the rationale. And you're virtually going to be making a gram stain because you're going to ask for, actually, you're not even going to ask for the gram stain because I determined that you're doing the gram stain. And I will set that up, take a picture of it, and then send the picture to you after the test incubates. And by the way, let's talk about that now. Let's come to here. Get out of here. Get out of here. Get out of here. Get out of here. Actually, I had to look up how to apply the virtual background. If you notice, it's now working. Apparently, they uploaded the Zoom and it lost the virtual background. I need to talk about the unknown rubric too. What was I talking about here?
The gram stain.
The gram stain, right. And that is, I said that you're going to be asking for a test, or I'm determining which test you run. And what you should do is look up these tests, which I need to make it available to you. In week 5 lab, the unknown tests. These are the unknown tests that you can run on your unknown number. Okay? And like for the gram stain, it goes through the different arrangement, and it's telling you about the cell shape and the growth arrangement there too. Let's look at that. So the gram stain, commonly done in microbiology, separating bacteria into gram positive or gram negative bacteria. You can also get the cell shape from the gram cell stain, and it's possible to get the cell arrangement. It tells you the cell arrangement here. However, it's warning you that in practice, it's usually difficult to determine. Let me shrink that down so I can read it. It's difficult to determine if the gram-positive cocci are in clusters or chains when viewed with the gram stain. This is because chains are broken apart during production of a smeared slide, and because cells tend to clump when brought together as artificial clumps, when water evaporates on a slide, when bacteria are taken from a agar plate and then put on a slide. Therefore, streptococci often look like clusters, and clusters can sometimes look like streptococci or some other thing. The message is, the take-home message, do not rely on the cell growth arrangement from the Gram stain. You need to perform the cell growth arrangement test instead. And then this goes through the procedure of the Gram stain. Put a small amount of cells from the sock culture on a drop of water on a slide, smear it over the entire slide. And when you do that, you can break up the streptococci. Allow the slide to air dry, and that can bring the cells together, making clumps of cells visible, or maybe tetrads or sarxinia, where they should be some other structure, cellular growth arrangement. Allow the cell to air dry, heat fix the slide, and then do the Gram stain. I'm not going to go through the Gram procedure here, but you can read about it. And then you can see some different pictures here. Gram-positive diplococci, you have to blow it up to see that, but there they are there. Staphylococci showing clumps. You can look there that looks like a streptococci, and it's not. Let me blow that up. Yep, didn't blow up right. Shrink that down one. Well, you got to blow that up, and I can't blow it up. There, kind of blowed up. That kind of looks like a streptococci, but this is actually staphylococci. It's just the problem of the Gram stain. And there's streptococci, and then either singlet bacilli or diplobacilli. Gram-negative bacilli, which I don't see there, but that's all right. All right, let's shrink that down. And then it tells you that you're getting your results. You can get your results immediately after doing the Gram stain. You just have to look under the microscope. Let's come back. See if I have a back there. Cell growth arrangement is the procedure you do for determining if your cells are clumped in clusters, like staphylococci, or if there are streptococci. And what you're doing here is your very gentle procedure where you take a bacterial smear, meaning you grow your bacteria in a liquid, so you're more likely to see the cellular growth arrangement of the bacteria, and then you put the bacteria on the slide in liquid, and then you look at the slide directly in the liquid. You can add stain if you want. And there you can see streptococci and diplococci there. So very gentle procedure that tends to keep the cellular growth arrangement intact. And those are both with staining. This takes at least a day to grow because you have to take your bacteria and grow it in a liquid culture. It may take 2 days to grow. Let's see what it says here. Or a day or two. Okay, so this one will require a day of incubation, maybe 2 days of incubation. And then let's go look at some other tests, like the MR test. Most of the tests will take 2 days of incubation. The MR test, on the other hand, takes 5 days to incubate. looking at mixed acid fermentation. And it says it's best to incubate for both 2 days and 5 days, but this is seldom required. And it tells you incubate for 5 days. And then run the MR test on the cells to determine if they're MR positive or MR negative. Any question about any of that?
So these are basically just walking us through the tests as if we were doing them in person. What you've done is you just lay them all out for us to go through the process and to get the results. Is that correct?
Yes, but you need to understand the test in order to write up the lab report. It's true that when you're doing the unknown notes, you may not need to understand the test, but when you do the lab report, you need to understand the test. And you will also need to know how long the test incubates, because every term I have students saying, "Where are my results?" And I will email them back, or actually in the unknown notes page, put in there, "How long does your test incubate? Look it up." Okay. So the point is, this test needs to be started at the very start of this project, because if you wait to the very end, if you're running, 3 or 4 additional tests after the 3 tests I send you, you will run out of time. In every term, I always have students who run out of time, in which case I tell them, okay, write up your lab report as far as you can, as best as you can, and just state you did not finish the test because you ran out of time. They don't get 100, but they will get some points. Okay? And it is best to begin this project at the start, because this project takes time. It'll take a minimum of 2 weeks, and that's only if you're lucky and you get the tests done in something like 2 tests or 3 tests. I've never had a student get it done in only 1 additional test, but I have had some students get it done in 2 additional tests after I give them 3 tests. Okay? So get started on this project right away, or you will run out of time, especially in the summer, because we're truncated on the time here. And I'm going to start this test today, meaning I will pick your unknown number, and you're going to pretend that you're streaking it out. I'm not going to tell you the number until I send out your results for the gram stain, the gelatin hydrolysis test, and the PR lactose test. But you're going to actually start the gelatin hydrolysis test today, because you will not get the results for a week. And then let me see what I've got in the instructions here. I might not have it yet. Yeah, I don't have it yet. Let me just go back to last term. Oh, what I'm trying to do is figure out which day you're doing the gelatin hydrolysis test today. No, I don't have it here. Just a second. I thought I had it there, but I put it someplace else.
I have a question. For this test, do we message you requesting it, or is something we automatically will get with the rest of the staying?
I'm going to automatically send you the first 3 tests. After that point, you have to request a test in your unknown notes pages. Okay, so the first test, I've already decided you are going to be doing the gelatin hydrolysis test because it takes a week, and that you're doing setting up today. And then you're going to set up the gram stain on next Sunday, but you're not going to look at the results until Tuesday. And then you'll be setting up the PR lactose test on Monday, and that test takes a day to run. And so I'll send all of the data for the gelatin hydrolysis test and the gram stain and the PR lactose test on next Tuesday. That's why I was looking up to see which days I was doing the test. Okay? And that's just to make it easier for me. I can send all this information as well as your unknown number in one email rather than send it 3 or 4 times in 3 or 4 emails. After that point, all test requests must be made in the unknown notes pages. If you ask a test in the email, I'll email it back saying I'm not honoring a test request in the email. And it's because I want all the information to be in one place for this project so that both you and I can easily look at the information and we know where to find it. It'll be in your unknown notes pages. So all test requests must be made in your unknown notes pages. Any question about any of that? All right, that means I got to set up the unknown numbers. I got to get that done today. I haven't done that yet.
I have a question.
Okay, what's your other question?
Can we request more than one test at a time or only one test?
You may request more than one test at a time. However, this project is using dichotomous logic. And if you request more than one test at a time, you will not be using dichotomous logic. You will then get a penalty.
Okay.
Okay? Occasionally at the end of the term, some students will request more than one test at a time, as well as some students who maybe do not understand what dichotomous logic is, and then they request more than one test, and then they get a penalty. If you request more than one test, I will ask you the question, are you sure you want more than one test? Because this project is supposed to be using dichotomous logic. Please confirm that you want more than one test at a time. I once had a student who simply asked for all the tests. She wanted every single possible test. And she actually did every single possible test. Okay? Obviously, she did not use dichotomous logic. All right, let's go to the instructions. Let me see if there's anything else I need to talk about that. Close that down, I think. So the project format, you're going to pretend to receive a broth culture. In reality, you're just going to get a number. You're virtually going to isolate the species. So I got that virtually. Oh, here it is there. You're virtually going to receive a broth culture containing A, meaning one bacterial species. So everything's done virtually. Using your one pure culture, run a series of tests to determine the identity of your species. You must use dichotomous logic. If you use not dichotomous logic, you'll be penalized. Shotgun logic is where you're just asking for a whole bunch of tests at a time. You're shooting with a shotgun, hoping that you hit the target. And you'll receive a loss of points for that. You must record your daily activities and your daily notes. That will be in your unknown notes pages. You then use your unknown notes pages to write up your unknown report. In your unknown report, it'll be worth 75 points. It will be an online report of your details for your thoughts and activities for each day of your unknown report. I'm getting a message saying the internet connection is unstable. So if you don't hear me, just ask me to repeat something. And if it breaks the internet, just stay logged in and I will re-log in. Please use 12-point font on all of the work that you are submitting, not just for the unknown report, but actually all of your work. If it's less than 12-point font, I should give you a penalty because I have a hard time reading it. Your daily descriptions should include the test you run, the information you hope to gain from the test, the observations you made from the test, that's your results, and then the conclusions from those tests. The conclusions are your interpretations for what the results mean. Like your observation is the test is yellow, or the test tube is yellow, or the test tube is something else like pink. If the test tube is yellow, your conclusion is that that test was positive, like in the PR inulin test. With PR lactose, you got to say whether it's actually AG or A or negative, meaning that one is not a yes or no answer. Do not omit data that'll make your project look better. I mean, I can just look up your data in the unknown notes pages if I need to. Drawings, photos, and tables are not required, but they are very helpful. You will get a picture and you may include photographs in your unknown report, but including them is not required. If you do include a drawing or photo or a table from the internet, you must completely label and discuss where you got that from. Otherwise, it's plagiarism. Each report entry must begin with the date. We don't really need a time for the online class, but your unknown notes pages will have a date for each entry. And let me see if I can call up a previous student's date. I've got to stop this sharing because I don't want you to see the student's grade on that, but I'll call up an unknown notes page with something here. I got to go to a previous class. There, you don't even know what class this is. For some reason, my internet is really slow now, too. It's probably a problem with the internet. Let's go. I'm going to remove the grade. That's why I don't want you to see this. There we go. So you sort of see the student. I can't do anything about that, but you won't see the grade. And now let me turn this on so you can see what I'm showing you. What the heck? Didn't let me share. Let's share. There we go. So you should see the page here. So this student looks like they made a unknown notes page where they started the very first day and then just added to it for each test they did. And it looks like this student didn't run it for that many tests. Yeah, they've only got 2 entries here. So the student either didn't finish the project or they got very lucky. Here's day 1 where they have the gram stain. And they stated that they see conchi and they're purple, and that means that it's a gram positive. There's a gelatin hydrolysis test. The gelatin in the tube remained liquid, so they're the bottom tube, which indicates to me that it's a positive result for this test. And then the PR lactose test is shown here. Don't know why they're not showing the negative control, but the tube is yellow, which indicates the presence of acid. And there are bubbles. Actually, there are no bubbles in this one. So they got that wrong. And then they did another test, which was actually on, they got the results on March 3rd, bile-esculin test. And there's the result. And that's esculin positive. There's blackening of the tube. That's the result. And the conclusion is that it's bile-esculin positive. And they actually identified the species here with just these tests. So this is a very lucky student. Okay, any questions? All right, let me stop the share and I'll put the grade back in that assignment. Lost my grade here, so I gotta look it up. Meaning I lost the copy and paste for some reason. It's not terribly important because the student has already gotten a grade out of this class. Oops. It overwrote the grade with, what the heck is that? Nothing? Yep. Well, I've lost it on the student. Sorry about that. Hopefully the student will never ask about it. All right. Any questions about any of that? If not, let's start sharing the grade there. I had it as a copy and paste, and for some reason, when I erased it, it didn't stay as a copy and paste. So I couldn't copy it back. Where were we? Oh, warning me that the internet connection is unstable again. You do not need to describe a step-by-step detail for each test you run. For example, if you run the gram stain, all you need to do is state that you did a gram stain. You do not need to go to the step-by-step cultures. You should put your thoughts and words in your report and use your own words. Your unknown report will be checked for plagiarism. It is mandatory that you include in your unknown report a table outlining the process or a figure that you use to identify your unknown organisms. An example is the table below here. Let me scroll to it. So this student initially, can you guys see this table?
Yes.
Someone there?
Yes, we can see it.
Yeah, great. Initially, they narrowed it down to the gram-positive coccus. So here are the possible species here. It then was catalase positive, narrowing it down to these four. They then did the PR mannitol test, narrowing it down to these three species. They then did the PR glucose test and got acid, narrowing it down to these two species. They then did the Simmons citrate test and it was positive, narrowing it down to Micrococcus varians. So this table is showing you the steps they used to identify it as Micrococcus varians. You must include either a table or a figure identifying how you obtained the species that you say it is. So that must be included in the report. You also must include a microorganism paragraph where you describe the microorganism that you have identified. So here they have identified Micrococcus varians, and you should discuss that in your microorganism paragraph. You only need a paragraph or two in length. Do not exceed one type page for this section. The report should be about 5 pages long. If it's longer than 6 pages or less than 4 pages, you will be marked down. I once had a student fairly recently who just copied a paragraph from somebody and then put it in your lab report. The plagiarism test noticed that and they were marked down for plagiarism. So you cannot simply copy the information from someplace. You need to write it in your own words. You must also give a literature citation page. You will have at least one citation, and that will be at the very least where you get your information that you're giving in your microorganism paragraph. If you have a figure there or a table that you got from the internet, you need to give that in your literature citation page as well. So this test will be going through a plagiarism test. I've never mentioned that plagiarism is important, and you will, at the very least, get minus 5%. I may give you a 0 on this project if you do a very bad job of plagiarizing. And plagiarism is important more than just in school. We once had a sitting senator who had to resign his Senate seat. And what he did was he plagiarized his graduate school work. I think it was his Master of Science thesis, where he plagiarized somebody. And that was obviously years later. And he had to resign his Senate seat. We also had a candidate running for the president of the United States, and this candidate had to drop out of the race because he plagiarized somebody else's speech, meaning he gave a speech and then didn't state that he got this speech from somebody else. Does anybody know who this candidate was? It's somebody you know. Does anybody know who this was? Nobody knows. It was actually the first time Joe Biden was running for the presidency. What he did was he was friends with this British politician, and the British politician made a speech and sent it to Joe Biden, and Joe Biden thought it was a good speech, and so he gave it. But he didn't state that I got this speech from so-and-so, and he was plagiarized it, and somebody caught him on it, and he had to stop running for the presidency. And I don't remember when that was, but it was long before 2020. I think it was even before 2000, but I don't remember. Any question about any of that? The point is plagiarism is important, and it's more important than just in school. Obviously, it is important in school. The grading criteria is listed below. I'm just going to skip to the grading criteria. I thought I had it here. I've got it on a separate page here. Oh, I'm not in the right place. Let me get there. Grading. Not the best because this is in a picture. So you'll be graded on the utilization of resources during isolation. If you have 0 excess media attempts, you'll get 10 points. If you have 1 media where it didn't give you any useful information, you'll get 8 points. You have 2 media where you didn't get any useful information, you'll get 6 out of 10 points, etc. Okay? If dichotomous logic is used, all tests were performed, were relevant, then you'll get, I don't think this is quite correct. Yep, it's out of 100 points. Let me look and see if I got this out of 75 points. There it is, the unknown report key. So if dichotomous logic is used, making sure it's out of 75 points, and there's no excess test, you get 10 out of 10. If there's 1 excess test, you get 8 out of 10. 2 excess tests, 6 out of 10. You use shotgun approach, you get 2 tests out of 10. If there's no logic used at all, you get 0 out of 10. If all the relevant tests were used, meaning you used all the tests you needed to identify your unknown, no tests were omitted, you'll get 10 out of 10. If 1 test is omitted, you'll get 8 out of 10. 2 to 3 tests omitted, 6 out of 10, etc. If the correct species are included in each step, you'll get 10 out of 10, meaning you've identified the correct species and all the different steps, the possible species at that point in time. 10 out of 10. If there's 1 incorrect species at one point in your list, then you get 8 out of 10. 2 to 3 incorrect species, 6 out of 10. Over 5 incorrect species, then 2 out of 10. If you do not state any species that are a possibility, then you'll get 2 out of 10. Okay? Because obviously none of them are correct. Completeness of report. If all the information presented is correct, 10 out of 10. If 1 thing is missing, you'll get 8 out of 10. 2 to 3 instances of missing information, 6 out of 10, etc. Effective and clear communication. Are your English, your grammar, your spelling, and your punctuation good? If you have excellent communication, 15 out of 15. A few errors, but logic is understood, 13 out of 15. Some errors, but logic is still understood, 11 out of 15. Some errors, and sometimes I can't understand your logic, you get 7 out of 15. Lots of errors, hard to read the paper, 1 out of 15. If your species you identify is correct, you'll get 10 out of 10. If the species you came up with is within the same group, same type, but it's not correct, you get 7 out of 10. If the species you come up with is outside your group, but the gram stain is correct, you get 5 out of 10. And if the species you come up with is incorrect and your gram stain is incorrect, you get 0 out of 10. for a total of 75 points. Any questions on the grading? All right, this is going through an example of dichotomous logic. If you understand dichotomous logic, you do not need to go through this last portion, but it goes through an example of dichotomous logic and then ask you a few questions. You do not need to turn in these questions. They are just for people who want a little practice with dichotomous logic. So it's another example. All right. Any questions about the project? I think that's it. If there's no questions, I'm done with the lab. On Thursday, we'll discuss Lab 6. Please remember you have a quiz this Wednesday. The quiz will cover Labs 3 and 4, Chapter 3, and the end of Chapter 2. Any questions? If not, I'm going to end the meeting here. All right, goodbye.